All right, welcome everyone. Today we are diving deep into the really clever and frankly kind of terrifying molecular strategies of Vibrio cholerae. In this first part, we're going to cover its basic profile and the incredible biology of its AB5 toxin. Then, in our next segment, we'll explore how this bacterium gets its hands on this pathogenic arsenal in the first place through something called horizontal gene transfer.
So, let's start with the central question, the one that really gets to the heart of it. Think about this. How does one tiny, single-celled organism manage to hijack our cellular machinery so completely that it turns our own cells into these unstoppable water pumps? To answer that, we've got to get to know the culprit.
So first things first, let's meet our antagonists, Vibrio cholerae. This isn't just any old pathogen. It's a true master of molecular sabotage. Understanding who it is the first step to appreciating its devastatingly effective strategy.
Okay, so what are we looking at? Vibrio cholerae is a gram-negative, comma-shaped rod. But a really key feature here is its single polar flagellum. Now this isn't just for, zipping around. It's actually a critical piece of its arsenal. It gives it the power to literally drill through the thick mucus layer in our intestines to get to the cells underneath.
Now what's really fascinating is that this species is incredibly diverse. We're talking over 200 different known serogroups. But, and this is the kicker, out of all those varieties, only two of them, O1 and O139, actually have the specific genetic toolkit required to cause those massive cholera epidemics.
And if we zoom in on that O1 serogroup, we see this really crucial evolutionary split. You've got the L-Tor biotype, which is causing the current 7th global pandemic, and it's a much tougher and stealthier adversary. It's way more resilient out in the environment, and critically, it causes a much higher rate of asymptomatic infections, which turns people into silent carriers.
So this table really lays it all out. Just look at that asymptomatic carrier ratio for the L-Tor biotype. Roughly 75% of infections don't even show major symptoms. That's huge. That, plus its ability to survive better out in the world, is exactly why it's such a persistent and successful global pathogen. It just keeps spreading silently.
Okay, so how do these specific strains actually do the damage? Well, it all comes down to their primary weapon, the cholera toxin. So let's break down the architecture of this thing, because from a molecular standpoint, it's both beautiful and incredibly deadly.
Cholera toxin is really the textbook example of what we call an AB5 toxin. It's this elegant two-part system. You've got one active A subunit, that's the toxic payload, and then you have 5 binding B subunits that form a ring. The whole design is built for one single purpose, precision delivery.
Yeah, you can think of it like a molecular Trojan horse, or maybe even a guided missile. Those 5 B subunits form a ring that acts as the guidance system, the key, if you will. It's designed to find one very specific lock on our cells. The A subunit, that's the warhead, the weapon, and it's completely harmless until it's safely delivered inside the target. So now let's get into the infiltration itself. How does this weapon so masterfully hijack our own cellular machinery? Because this isn't some brute force attack, it's a super subtle process that uses the cell's own most basic functions against it. And so, the attack begins. That B subunit key finds its lock, a molecule called the GM1 ganglioside receptor, right on the surface of our intestinal cells. And it doesn't just bind, it latches on with what the sources call extraordinarily high affinity. Once it grabs on, it is not letting go.
So with the toxin latched on tight, the cell gets completely fooled. It sees this thing on its surface and just does what it normally does. It starts a process called endocytosis, basically just swallowing the entire complex whole. The cell itself literally pulls the enemy right past its main defenses.
Now, once it's inside, you'd think it would get broken down, right? Nope. Instead, the toxin pulls off this absolutely brilliant move. It hijacks the cell's internal mail system and travels backward. It's a process called retrograde transport. It goes from the Golgi apparatus all the way back to the cell's protein factory, the endoplasmic reticulum.
We're about to see what happens inside the ER in just a moment. But hey, if you're finding this deep dive valuable, please do me a quick favor and hit that like and subscribe button. It really helps us make more custom medical content just like this for you. Okay, so the toxin is now hidden deep inside the endoplasmic reticulum, and this is where the A subunit is finally set loose to pull off the final irreversible biochemical coup. This really is the point of no return for the cell. So that active part of the weapon, the A1 fragment, is now free inside the cell's command center, and it has one very specific target,
a protein called GS-alpha. The best way to think about GS-alpha is as a critical light switch that controls a really important cellular pathway. The toxin sabotages this switch using a chemical reaction called ADP ribosylation. It basically modifies the GS-alpha protein in such a way that it's like taking a pair of pliers and just snapping off the off button. That switch is now permanently, irreversibly locked in the on position.
So with that switch jammed on, the GS-alpha protein just non-stop stimulates an enzyme called adenylate cyclase. And this enzyme goes into absolute overdrive, just churning out a crucial signaling molecule called cyclic AMP, or CAMP. Before you know it, the cell is completely flooded with the stuff.
And this massive flood of CAMP is what kicks off the final devastating cascade. It turns on another enzyme, protein kinase A. And that enzyme's job is to force open a specific ion channel called CFTR. So now this channel is just stuck wide open, leading to this massive, unstoppable flood of chloride ions pouring out of the cell. And as we know, where chloride goes, sodium and water have no choice but to follow.
And that right there, that's the ultimate consequence of this incredibly elegant molecular attack. Just by permanently locking open a single type of ion channel, the bacterium has completely reversed the cell's normal function. It has turned a cell that's supposed to absorb nutrients into a hypersecretory one. It has literally turned our own cells into tiny, non-stop water pumps.
All right, now continuing with the TCP pilins and bacteriophage horizontal gene transfer. We're about to get into a really fascinating story, one that's basically about genetic theft and how this bacteria uses a very special tool to literally steal its deadliest weapon. So, let's start with this big question, right? You've got all these Vibrio bacteria out in the world, and most of them are totally harmless. So how does one of them suddenly get its hands on the cholera toxin? It's not born with it. The answer? Well, it's a story of molecular piracy. Okay, so the first piece of this puzzle is a really crucial bit of equipment. I want you to picture a grappling hook. That's pretty much what we're talking about here. It's called the Toxin-Coregulated Pilus, or, much easier to say, TCP for short. So what is this TCP thing? Well, if you could zoom way, way in on the bacterium's surface, you'd see this tiny, hair-like thing sticking out. And let me tell you, it is absolutely essential for what comes next, colonization. I mean, without this thing, Vibrio cholera has no way to get a foothold inside the intestine. It just can't cause the disease. You could almost think of it like a three-step tactical operation. First, the bacterium has to punch through the protective mucus layer that lines our intestines, which is not easy. Then, once it actually gets to our cells, the epithelial cells, that's when it shoots out the grappling hook, the TCP pilus, to latch on super tight. And that moment, that adhesion, that's the first critical move to setting up shop. But here's where it gets really clever. That grappling hook, it has a secret double life. It's not just for hanging on for dear life. It's also a doorway for the second key player in this whole drama, a viral messenger known as the CTX phage. So just to be clear, we know the TCP pilus is the absolute essential tool for the bacterium to attach itself. But like I said, it's got this secret second function. And this is where the plot really thickens. It's almost like something out of a spy movie. Because here's the big twist. That grappling hook, it is also the specific receptor, the lock on the door, if you will, for a virus called the CTX bacteriophage. So the very tool the bacteria evolved to survive is also its greatest vulnerability to this viral invader. How wild is that? Okay, so what is this CTX bacteriophage, or CTX-5? It's a type of virus that only infects bacteria. But here is the absolute kicker. It carries the genetic blueprint for the cholera toxin. Think about what that means. The genes that make this disease so deadly, they aren't native to Vibrio cholerae at all. They are literally delivered, like a prepackaged weapon, by this viral messenger. So you might be wondering, how does one single bacterium latching on turn into a full-blown infection? Well, it's all about establishing what we call a beachhead. And what exactly is a beachhead in this context? Get this, that same TCP grappling hook doesn't just stick to our cells, it also sticks to other Vibrio bacteria. It's like they're holding hands. This lets them all clump together, forming this protective little neighborhood called a microcolony. It's exactly like an army landing on a hostile shore and securing a position so they don't all get washed back out to sea. And you really can't overstate how important this is. Without that beachhead, Vibrio cholerae just can't build up its numbers. It can't create a big enough population to produce enough toxin to actually make someone sick. It's that simple. In this battle, colonization is absolutely everything. So this whole process, it leads us to the really profound evolutionary strategy that's going on here. You could really call it the art of the steal. Now, the proper scientific term for this is phage transduction, and it's a perfect example of something called horizontal gene transfer. Normally we think of genes being passed down from parent to child, right? That's vertical. But bacteria, they can pass genes sideways to their neighbors, and in this case, they use a virus as the delivery truck. It's an incredibly powerful way for them to pick up brand new traits almost instantly. And this has profound implications. Just think about it. It means the toxin genes aren't locked into one type of bacteria. They can theoretically be transferred to other totally harmless Vibrio strains. This isn't some freak one-time event. It's a repeatable mechanism for creating new pathogenic threats out of thin air. So if there's one thing to really get from all this, it's this. Vibrio cholerae doesn't just have its weapon, it acquires it. That means pathogenicity, the ability to cause disease, is a transferable trait. A harmless bacterium floating around today could become a deadly pathogen tomorrow just by getting infected by the right virus. And that leaves us with a pretty heavy question to think about, doesn't it? If virulence, the ability to make us sick, can be passed around so easily, so elegantly, between bacteria using viruses as little couriers, what does that mean for the future, for the emergence of brand new diseases? It paints a picture of this constant modular evolution happening all the time, right under our noses. All right, let's bring it home with a clinical pearl. Why do we care about all these molecular details? Well, it's simple. If we know that the TCP pilus is the doorway that lets the toxin gene in, we can start designing therapies that block that doorway. If you can jam the lock, the dangerous package never gets delivered. Hey, if you enjoyed this deep dive into some pretty complex medical science, please do us a favor and hit that like and subscribe button. It helps us make more of these explainers for you.